DNA fingerprinting, classical method. Also known as Rflp-based DNA fingerprinting. Rflp equals restriction fragment length polymorphism. Old DNA fingerprinting relied on detecting differences in the length of DNA fragments produced by restriction enzyme digestion at specific sites within variable number tandem repeat, VNTR, regions of DNA. VNTRs are longer repeat sequences, 10 to 100 base pairs, that vary in number among individuals. These variations produce fragments of different lengths forming a unique banding pattern, the fingerprint. Principle: restriction enzymes cut DNA at specific recognition sequences. Differences in repeat numbers, VNTRs, cause fragments of different lengths. When these fragments are separated and visualized, they produce distinctive bands for each person. This is the DNA fingerprint. Steps of DNA fingerprinting, Rflp method. Step 1. Sample collection. DNA was extracted from biological samples such as blood, semen, hair roots, or tissue. Note: old methods required large, good quality DNA, degraded samples did not work well. Step 2. DNA extraction. DNA was purified using phenol chloroform extraction and alcohol precipitation. Step 3. DNA digestion. The purified DNA was cut into fragments using restriction endonucleases, example EcoRI, HYDE-III. These enzymes recognize specific sequences and cut DNA, generating fragments of varying lengths depending on the VNTR regions. Step 4. Gel electrophoresis. The DNA fragments were loaded onto an agarose gel and separated by electrophoresis based on size. Smaller fragments migrated faster than larger ones. Step 5. Southern blotting. The separated DNA fragments were transferred from the gel to a nylon or nitrocellulose membrane, using capillary action. This made the DNA accessible for hybridization. Step 6. Hybridization with DNA probes. The membrane was exposed to radioactively labeled DNA probes complementary to the VNTR regions. These probes bound, hybridized, to the matching DNA fragments. Step 7. Autoradiography. The membrane was placed on X-ray film. The radioactive signals from bound probes exposed the film, producing dark bands corresponding to specific VNTR fragments. Each individual showed a unique pattern of bands. This was the DNA fingerprint. Output: on the developed X-ray film, each person's DNA showed a unique set of dark bands. Related individuals showed some common bands, inherited VNTRs. Advantages: feature, description, highly specific, accurate when high-quality DNA is available. First scientific method for human identification. Revolutionized forensic science and genetics. Limitations. Problem. Explanation. Large DNA quantity needed. Required micrograms of high-quality DNA. Time-consuming. Several days to weeks for results. Complex procedure. Involved radioactive probes and manual analysis. Not suitable for degraded samples. Could not analyze old or minute forensic traces. Health and safety issue. Used radioactive materials for probe labeling. Comparison: classical vs modern DNA fingerprinting. Feature: old, Rflp based. Modern, STR-PCR based. DNA quantity required: high, micrograms. Very low, nanograms. DNA quality: must be intact. Works even with degraded DNA. Main region analyzed. VNTRs, 10 to 100 BP repeats. STRs, 2 to 6 BP repeats. Detection method. Radioactive probe plus X-ray film. Fluorescent dyes plus computer. Time required. 1 to 2 weeks. Few hours. Automation. Manual. Fully automated. Result format. Banding pattern on film. Digital electrophoregram. Thanks for watching.